Today we start with some ideas about the calculation of crop productivity. In this class we will talk about the different process of growth and development of the crop, and also the very useful concept of thermal time and the calculation of the cycle duration. We have to clearly distinguish between growth and development, because growth is a quantitative change that implies the increase in size of mass or the plant or the crop. It is associated with the use of resources. On the other hand, development is a qualitative change in the form or in the function of the plant. The crop cycle is separated by given characteristic phases, and the duration of these phases depend on environmental signals like temperature, light, or photoperiod. We can distinguish between morphological development, for instance, the appearance of leaf on the stem of the plant, or phasic or phenological development, which is related to the duration of the phases in the crop cycle. But we have to emphasize the idea that growth means a quantitative change, while development means a qualitative change in the plant. The different stages in the crop cycle for an annual crop are as follows. First, we have the sowing, which means when we put the seeds or the propagules for the crop on the soil. Second is the germination, the appearance of the radicle in the seed. Third would be the emergence, the appearance of the plant over the soil. Fourth would be a juvenile phase. This is a phase when the crop is not able to detect environmental signals that trigger the start of reproductive growth. And during this juvenile phase, we have initiation of leaves. We have the initiation of small microscopical structures that will be in the future the leaves of the plant. After the juvenile phase, we have floral initiation. This is the formation of the primordia of the reproductive structures. Later, we will have flowering, followed by physiological maturity, at which time the biomass of the harvested organ is maximum, and finally harvest maturity, when the conditions are best for mechanical harvesting. Here we have an example of sunflower development. Here we have the emergence of the crop. We see the two cotyledons. Here we have a V2, which means vegetative stage, and the number 2 is used to indicate the number of true leaves. We have the cotyledons plus a couple of leaves. Here we would have a V4 with 4 true leaves apart from the cotyledons, and so on. The number of leaves can go up to 30 something or something like that. After flower initiation, we would observe the R1 stage, which is already a reproductive stage. It is also called the star or bud visible, where we see a kind of a star which would be hiding the small microscopic structure that will become the head of the sunflower plant. Here the head has already started to grow. The head will be growing for different reproductive stages, and at R5 the sunflower plant starts anthesis, in other words flowering. Finally, the plant would be ready for mechanical harvesting at R9. We said that at flower initiation we have a small microscopic structure in the plant that will become the reproductive organ. That occurs at the jointing stage in the case of wheat, where we already have stem growth. And inside the plant we see a structure like this one. This is a microscopic structure that will become the spike of the wheat plant. Look at here the scale. This would represent 1 millimeter, so this is around 2 millimeters in diameter, and this is hidden inside the stem of the plant. We already have seen the different stages of the crop for annual species. In the case of perennial species, we have a more complex situation. First, we have a long juvenile phase if the plants are propagated using seeds. That would mean that when we plant perennial species, in particular trees, from seeds, it will take some years before the plants are able to flower. However, for trees it is very common to use vegetative propagation from cuttings, and these cuttings are not juvenile, so that reduces the time until flowering. In any case, when we establish the plant, we will have annual cycles similar to those of an annual plant. Here we have an example of vegetative propagation of cuttings with olive plants. Here we are growing olive cuttings in perlite. After some time, well, this is grown in a shade house with high humidity and warm temperature. After some time we have production of roots for each of the cuttings, and they are transplanted into small pots where they are grown in shade houses for 1 to 2 years, after which they are transplanted to the field, like here. We can distinguish between two types of perennial species. First, we have deciduous species, which encompass most fruit trees and vines. They lose their leaves in the autumn in response to cold temperature and or short photoperiods. After the leaves are lost, the buds stay dormant during winter, so they are not sensitive to environmental signals for bud break. After some time with cold temperatures that are called chill requirement, bud break will occur in the spring. After that, vegetative and reproductive growth will overlap. In the case of evergreen species, which include citrus, the citrus species, plus olive trees and tropical fruit trees like the mango, the leaves stay in the plant for 2 to 3 years. In some cases, the tree may stay dormant during the winter, so there is absolutely no vegetative growth during the winter. The amount of overlap between reproductive growth and vegetative growth depends on the species. We have a species where the flowering occurs before leaf growth. Will be, for example, peach, like in this picture. In other cases, like in grapevine, the flowering will occur long after leaf growth starts. The overlap between vegetative and reproductive growth depends also on the date of harvest. For instance, cherries are harvested in late spring to midsummer, so they have a rather early harvest, and after that we will have only vegetative growth. In the case of grapevines, we will harvest much later, almost in the autumn, so we will have a lot of overlap between vegetative and reproductive growth. The main factors affecting phenological development are temperature and photoperiod. In the case of temperature, we have a positive effect of high temperatures on development rate, and this is related to the concept of thermal time, but we have also a response to cold temperatures, which is included in the concept of vernalisation. Apart from temperature, the plants require also an adequate water content for germination to occur. The seeds need to get hydrated before germination occurs, and also in some cultivars of some species we have a response to the photoperiod. The thermal time concept is related to a direct effect of temperature or temperatures on development rate. In this picture, we plot development rate versus temperature, and we have that in reality we would have something like the broken line that can be approximated by two straight lines like here. The straight lines intercepts the temperature axis at Tb, which is the base temperature for development, and goes to a maximum value for T0, which would be the optimum temperature for development. For much higher temperatures, the rate of development will decrease. Thermal time is the sum of the temperature above base temperature for a given phenostage. If thermal time for a given phenostage is constant, then we can deduce its duration using this equation. This equation, d is duration, is equal to the thermal time for the phenostage divided by temperature minus base temperature. For instance, for the emergence of a wheat crop, we require a total thermal time of 110 growing degree days. This is the unit for thermal time, growing degrees days, with a base temperature of 0 degrees. According to that, if we have a temperature of 10 degrees, we can calculate the duration of the period from sowing to emergence, dividing 110, which is the total thermal time required, divided by the temperature minus the base temperature, which is 0, and that is equal to 11 days. If we are growing the crop at 15 degrees, then we apply the same equation, 110 over 15 minus 0, and that is equal to 7 days. Therefore, this equation allows the calculation of duration of phenostages as a function of the temperature of the air. In this table that you can find in the book of Villalobos and Ferreres 2016, we see the base temperature and the total thermal time required for the phase sowing to emergence for different crop species. We see that the base temperature can be as low as 0 in the case of winter cereals, wheat or barley, and it can be as high as 15.5 for melon or watermelon. Typically, the legume crops, pulses, have rather low base temperatures, and they will be adapted to autumn or winter sowing, with base temperatures usually below 6 degrees. On the other hand, we have species more adapted to spring-summer sowing, like maize, base temperature 8, millet from 8 to 13, peanut, etc. Many horticultural crops, like pepper, will require rather higher temperatures. Tomato 9.3, etc. So the range in base temperatures might go from 0 to around 15 degrees. Interestingly, these values will be related to the base temperature of other components in the agricultural ecosystem, as we will see later. If one is interested in finding the typical crop cycle duration for different crops, we can use the Appendix 11.1 of Villalobos and Ferreres 2016, where we find this type of data for any crop species and different climate types, Mediterranean, continental, etc. And for specific sowing dates, we find the duration of the 4 phases used in the FAO crop coefficient curve method. This would be the initial phase, the rapid growth phase, this would be the maximum crop coefficient phase, and that would be the declining crop coefficient phase. We see also in this table the total duration of the crop in days. As we said, base temperature for crops may lie between 0 and around 15. It is interesting that for insects of different orders, we can see that the base temperatures are related to the crop temperatures. We see here 5 insect orders: Coleoptera, Diptera, Hemiptera, Hymenoptera, and Lepidoptera, and we see the average base temperature for development that are between 8 and 10 degrees. The minimum and maximum values are from 0 to around 19 degrees, which overlaps mostly the base temperature for crops. In this table, you can see also the total thermal time, called here degree day requirement, for the time from oviposition to adult eclosion for the different order of insects. Here we see that we require from more than 200 growing degree days to more than 500 growing degree days on the average for that period to occur. We said also that there is a response to low temperature which is called vernalization. This process, as well as photoperiod, are mechanisms that allow the avoidance of extreme conditions like frost or drought during flowering. In this graph, we see the reductions in grain number due to stress for different times of the crop cycle, growing from sowing to anthesis to physiological maturity. We see that for wheat, the crop is more sensitive to stress before anthesis, while for soybean, the crop is most sensitive after anthesis, while in the case of maize, anthesis is the most sensitive period of growth. In any case, for the three crops, the time around anthesis is highly sensitive for the determination of grain number, and grain number is usually the main limiting factor for yield. Therefore, it is important for the crop to avoid stress during this period. That can be achieved by using shorter cultivars, and in this example, in this table, we see the thermal time to flowering for different wheat cultivars in Australia from the late 1800s to the end of the 20th century. It is clear that the growing cycle, the growing season length has decreased with the breeding programs, and that allows better conditions during flowering, because in this case, longer time to flowering means entering under summer conditions which have a higher risk of drought. To avoid bad conditions during flowering, the vernalisation will allow the crop to avoid the occurrence of reproductive growth until an amount of cold has been accumulated by the plants. In other words, we have a requirement of low temperatures before flowering can occur. This is a typical requirement in winter cereals, but also in trees. The cold is accumulated since seed imbibition. After the seed is hydrated, it will accumulate cold. Apart from that, it's a reversible process, which means that if we have high temperatures, devernalization might occur. The requirement of vernalization depends on the cultivar, so we might distinguish winter cultivars that have a requirement of vernalization versus spring cultivars that do not require any vernalization. For instance, in the case of southern Spain, most wheat cultivars are spring-type cultivars. Both for annuals and trees, there is a large variability in the requirement of cold, in the vernalization requirement. The effect of vernalization on flowering is illustrated in this picture from the words of Lysenko in the old USSR, where they tried to sow winter wheat in spring on several years, and if the number of chilling days, if the amount of cold was not enough, the plants would not set flowers. Here we see what is the effect of a different length of cold conditions for the seed. The wheat seeds were embedded under cold conditions for different periods, and if the seeds were under chilling conditions for more than 1 month, like here 36 to 51, the plants would flower later. If the chilling period was smaller than 30-some days, the plants would stay vegetative and would not set flowers. In the case of trees, we usually talk about chilling hours for the vernalization requirements. This chilling is required for the buds to produce later flowers. And we have a large difference among species and cultivars. Typically, apple or cherry trees have a high requirement of chilling hours, while other species like almond have low requirement. In this map, we see the amount of chilling hours calculated as number of hours below 7 degrees. We see that typically most of the Iberian Peninsula has more than 1,500 chilling hours per year, and only in areas in the south and on the coast we see that the chilling hours are below 1,000. So on most areas of the Iberian Peninsula there will be no problems for chilling any year. But in the warmest areas and in the coast, in the south and in the coast, we might have chilling problems, lack of chilling hours during specific years. In parallel to the requirements of vernalization to avoid bad conditions during flowering, because if we have a very early flowering, the risk of frost damage will be high, and using vernalization we avoid early flowering. We have also photoperiod as another factor to avoid very early flowering. The response to photoperiod actually is a response to the duration of the night. We can distinguish between long day plants and short day plants. Long day plants like winter cereals will accelerate their development rate under long day conditions, with long days. On the other hand, short day plants like summer cereals or soybean will show their maximum development rate under conditions of short days. There is a lot of genetic variability within the species. So we find, for a given species, we can find plants that respond or not to photoperiod. In the case of soybean, the response to photoperiod allows having soybean cycles ranging from 90 to 190 days. It is important to stress the idea that the response to photoperiod is quantitative in the case of agricultural plants. And quantitative means that things go faster or slower for different photoperiods, but they do not stop completely, which would be the case of a qualitative response. Photoperiod also affects the leaf senescence in trees. Here we have an example of an experiment where the photoperiod was manipulated for Papaver somniferum here in Cordoba. We see plots with crops that had an extended photoperiod using artificial light. At the end of March, those plants that were supplemented with light with a photoperiod of 24 hours were already flowering, while plants under the natural photoperiod, which in that period is around 12 hours, were still on vegetative growth. Apart from phenological development, which means the duration, or which relates to the duration of stages in the crop cycle, morphological development is related to the appearance or disappearance of the different organs of the plants. The meristems might follow different fates. They may remain dormant, produce leaves, branches, or flowers, or produce reproductive structures. The organs appear or die, and that leads to the plant architecture. This is in response to temperature, radiation, and light quality. The plant architecture is very important in agronomy because it affects the lodging risk of the crop, the harvestability of the crop, the competition with weeds, the responses to herbivory. So what happens if the plant is cut at different heights, and the distribution of light and chemicals that we might apply. The strategies of meristem allocation imply the existence of trade-offs. We win some and we lose some. For instance, the strong apical dominance, and we see here in the picture in the left, we see a plant with different stems, would be a maize plant with different stems from the base, with tillering properties, as compared to a modern maize plant with a single stem and a single cob. If we have a strong apical dominance, which would be the case of this modern maize plant, the lateral buds stay dormant, so no branching occurs. That favors the competition for light because it will favor the height growth of the plant, and it will recover better after herbivory. On the other hand, it will imply a reduced growth capacity of the plant and a reduced capacity for filling gaps for this plant as compared to a more branching type plant. The morphological development is associated or is related to process like photomorphogenesis, which allows the plant to detect the neighbors and their properties. The plants can detect light quality. They can measure the red-far red ratio by the phytochromes, and that triggers change in morphology. For instance, with a low red-far red, that means that we have plants on top or on the sides. Why? Because plants absorb red better than far red, so if the plants are transmitted through, if the light is transmitted through a canopy, the amount of red is reduced much more than the amount of far red, so the ratio will be lower. That will cause the plants to respond with longer internodes, using more or distributing more carbon to the stems, reduce leaf growth, and reduce branching. That favors the growth in height and the competition against the neighbors. We have already analyzed the process of development, either phasic or morphological development, and now we'll dedicate some time to analyze the process of crop growth. As we said, crop growth is related to the use of resources. In this picture, we see that we use resources from the air, from the atmosphere, radiation and CO2, and from the soil, like nutrients or water. The rate of use of radiation or CO2 depends on a set of regulators, for instance, day length, temperature, light quality, and biotic factors. And the rate of use of soil resources depends also on temperature, but also on soil compaction, salinity, and also on biotic factors. The growth will depend on the amount of resources captured by the crop, but also on the velocity or the efficiency of use to transform these resources into plant biomass. Growth usually follows sigmoid curves, which means S-shaped curves. In this example, we see the time course of biomass in leaves and grain of a maize crop in Florida. We see that the leaf growth ends when the grain growth starts to become rapid. So we have clearly, in the case of maize, a determinate growth, which means that the reproductive growth is clearly separated from vegetative growth. We can measure growth by sampling the crop at different times, measuring either the dimensions, the length, the area, or the volume of plant organs. We can sample plants and measure the dry mass expressed in grams per square meter, kilograms per hectare, or tons per hectare. And for roots, we measure the root length density, which is the length of roots per unit volume of soil. We can express the crop growth rate as the simple definition is the increase of biomass per unit time, and relative crop growth rate would be the ratio of crop growth rate and biomass. In this graph, we try to summarize the two factors associated to growth, which are the use of resources and the efficiency in the use of resources. We start by a maximum level of biomass, which is obtained under unstressed conditions, following this straight line. So the total amount of resources, which can be water or radiation or nitrogen, are maximum. If we reduce the amount of resources captured by the crop, we would have this amount of biomass limited by resources. But we have followed the same line with the same slope, so we are still under conditions of lack of stress. If the stress increases, we can reduce biomass production to this point, where the amount of resources have been limited, like here, but also the slope of the relationship follows this discontinuous line, the stress line. So, for most situations, the amount, the productivity of the crop, the growth of the crop is limited by the amount of resources captured by the crop. and if the level of stress is higher, we will also reduce the efficiency in the conversion of resources to crop biomass. Finally, an interesting point about the main effect on temperature on crop production. In general, the main effect of temperature is related to the duration of phenological development. So, in temperature environments, late sowing shifts the crop cycle to warmer conditions, shifts the crop cycle to the summer. Development proceeds faster, the period available to capture resources and growth is reduced, and biomass at maturity is normally lower. In other words, in general, higher temperatures in temperate environments means a shorter season length and therefore less biomass produced at maturity. This is illustrated for this experiment with faba bean. Here we have the amounts of intercepted PAR, biomass or radiation use efficiency, and the cycle duration. Let's only look at the biomass, which is the green line, and the cycle duration, which is the purple line. We see that the cycle duration is reduced as we delay the sowing date. This is almost coincident with the variation in biomass. So biomass tends to reduce with a later sowing date, with the exception of this first point that corresponds with very cold, extremely cold conditions for part of the growing season. So in general, there is a parallel reduction in biomass production as the crop cycle gets longer, with the exception of conditions of very low temperature, which will limit directly crop biomass with a reduction in photosynthesis.